Veterinary drugs are an important component of animal healthcare, helping to prevent and treat a wide range of diseases and conditions in domesticated and wild animals alike. These drugs are derived from a variety of sources, including natural compounds found in plants and animals, as well as synthetic compounds developed in laboratories. The production of veterinary drugs involves a complex series of biological processes, including fermentation, extraction, purification, and formulation. These processes are critical to the development of safe, effective, and high-quality veterinary drugs that can be used to improve the health and welfare of animals. Fermentation. Fermentation is a key biological process in the production of many veterinary drugs. In fermentation, microorganisms such as bacteria or fungi are grown under controlled conditions, and the resulting biomass is harvested and processed to extract the desired compounds. One example of a veterinary drug produced through fermentation is the antibiotic penicillin. Penicillin is produced by the fungus Penicillium chrysogenum, which is grown in large-scale fermentation tanks. The fungus is fed a nutrient-rich broth that provides the necessary carbon and nitrogen sources for growth, and is allowed to ferment for several days. After fermentation, the biomass is harvested and processed to extract and purify the penicillin. Extraction. Extraction is another important biological process in the production of veterinary drugs. Extraction involves the separation of the desired compound from the biomass or matrix in which it is found, using solvents or other methods. One example of a veterinary drug produced through extraction is the anti-inflammatory drug aspirin. Aspirin is derived from salicylic acid, which is found in the bark of willow trees. The bark is harvested and processed to extract the salicylic acid, which is then chemically converted into aspirin. Purification. Purification is a critical step in the production of veterinary drugs, as it ensures that the final product is safe, effective, and of high quality. Purification involves the removal of impurities and contaminants, using a variety of methods such as filtration, chromatography, and distillation. One example of a veterinary drug that requires extensive purification is insulin. Insulin is a hormone produced by the pancreas, and is used to treat diabetes in animals and humans. Insulin is purified from animal pancreases using a combination of chromatography and crystallization, and must be rigorously tested to ensure that it is free of contaminants and is of consistent potency. Formulation. Formulation is the final step in the production of veterinary drugs, and involves the preparation of the drug in a suitable form for administration to animals. Formulation can involve the preparation of tablets, capsules, injectable solutions, or other dosage forms, depending on the needs of the animal and the properties of the drug. One example of a veterinary drug that requires careful formulation is vaccines. Vaccines are used to prevent infectious diseases in animals, and must be formulated to ensure that they are safe, effective, and stable. Vaccines can be formulated as inactivated or live-attenuated viruses or bacteria, and may require the addition of adjuvants or other components to enhance their efficacy. In addition to these biological processes, the production of veterinary drugs also involves strict quality control measures and regulatory oversight. Veterinary drugs must be produced in accordance with good manufacturing practices, GMP, which ensure that the drugs are consistently produced to a high quality standard. Regulatory agencies such as the U.S. Food and Drug Administration, FDA, and the European Medicines Agency, EMA, oversee the approval and registration of veterinary drugs, and ensure that they are safe and effective for use in animals. One challenge in the production of veterinary drugs is the need to develop drugs that are safe and effective for use in a wide range of animal species,